Hello, guys. I know it's quite dark over here, but anyway, possible to watch. So, today's lesson is about viruses. Let's go.
For reference, in 1892, the Russian scientist Ivanovsky studied the specific characteristics of the pathogen called tobacco mosaic, which occurs in tobacco plants. Viruses that cause this disease have the ability to pass through the bacterial filter. As a result, a healthy tobacco plant can be damaged by the filtered liquid. A few years later, Leffler and Frosch concluded that the causative agent of foot and mouth disease in pets also passes through the bacterial filter. Finally, in 1917, F. de Erel discovered the bacteriophage virus that infects bacteria. Viruses are the causative agents of influenza, rabies, jaundice, encephalitis, rubella, and other diseases. The interesting thing is that viruses cannot be found and multiply outside the cell in a free and active state. Viruses such as bacteriophage and adenovirus have DNA. Viruses that cause diseases such as encephalitis, measles, rubella, rabies, and influenza have RNA. Viruses consist of nucleic acid and proteins that form the viral envelope surrounding it. The virus shell is called a capsid. Interaction of viruses with cells. Viruses can accidentally enter the cell through pinocytosis, vacuoles formed from the fluid in the intercellular environment. But usually before the virus enters the cell, it binds to a special protein receptor on the cell surface. This connection is carried out by special proteins on the surface of the virus. They have the ability to recognize a sensitive specific receptor on the cell surface. The part of the cell attached to the virus merges with the cytoplasm and becomes a vacuole. The shell of a vacuole consisting of a cytoplasmatic membrane fuses with another vacuole or nucleus. In this way, the virus can spread to all parts of the cell. Accumulation of virus particles causes them to leave the cell. This process occurs in the form of an explosion in some viruses. As a result, the cell dies. Other types of viruses divide in a similar way to budding. In this case, the organism cells remain viable for a long time. Bacteria virus. The entry of bacteriophages into the cell is slightly different. The thick cell membrane of bacteria, like animal cells, does not allow it to penetrate into the cytoplasm together with the virus attached to it. Therefore, the DNA or RNA located at its head is pushed into the bacteriophage cell using a hollow root. The genome of the bacteriophage enters the cytoplasm and the capsid remains outside. In the cytoplasm of the bacterial cell, the genome replication of the bacteriophage begins and the protein is synthesized and its capsid is formed. After a certain period of time, the bacterial cell dies. Macrophage viruses are released. The origin of viruses. Viruses are autonomous genetic structures and cannot develop outside the cell. It is assumed that viruses and bacteriophages are special genetic elements of the cell that have evolved together with cellular forms of life. Currently, viruses are widely used in the field of genetic engineering.